Exscalate4Cov High-Performance Computers Against Coronavirus. Fighting climate change, managing energy supplies, creating new drugs in a short time. Nowadays, it is possible to exploit the capacity of high-performance computing to process large quantities of data, big data, and to find answers to current challenges. Coronavirus is one of these challenges. The Exscalate4Cov project was created to face this challenge in order to accelerate the research of a medicine able to inhibit its progress. As opposed to vaccines, which give immune protection also to people who are not affected by diseases, antiviral medications are administered to treat a condition and reduce its symptoms. But how does a high-performance computer actually find an effective drug against coronavirus? First of all, it is important to understand what a virus is. A virus is a non-autonomous organism which infects other organisms to reproduce. These organisms are defined hosts and may be humans, animals, or bacteria. There are several types of viruses in nature, and they can infect in different ways. However, they all show the same basic structure. Viruses consist of a protein capsid which contains genetic material, DNA or RNA. The coronavirus is not an exception. Like other viruses, the coronavirus consists of a protein capsid. Inside this capsid, there is its genetic material in the form of RNA molecules. All over the capsule's surface, there are proteins called spike proteins. As well as giving the crown shape typical of this virus, the spike proteins are crucial because they allow it to get into human cells. How does the coronavirus infect human beings?
Using spike proteins, the coronavirus binds to ACE receptors scattered all over the surface of human cells.
The interaction between proteins on the virus's surface and proteins on the human cell's surface promotes the spread of the virus's genetic material inside our cells. Once inside, the viral RNA interacts with the ribosomes of our human cell. Ribosomes are macromolecular complexes able to read information given by viruses' RNA molecules and to translate it into viral proteins. Once produced, viral proteins will build the capsid, which covers the virus, and create new copies of viral RNA. Eventually, capsid proteins and RNA molecules merge to create new particles able to infect other human cells. How can a drug stop the virus? Researching an antiviral drug means identifying molecules able to engage with proteins that are used in the production of new viral particles. The activity of these proteins can actually be affected by using smaller molecules able to weaken their usual performance. When the activity of viral proteins is stopped, the replication of the virus is stopped as well. Here, the European project Exscalate4Cov enters the game. Exscalate4Cov is a project created by a public-private consortium supported by the European programme Horizon 2020, including 18 research organisations all over Europe. Among these, there is also the Big Data Association. The association consists of the main Italian research institutes and universities of the Emilia-Romagna region. The starting point of the project is a database containing more than 500 billion compounds. Using the processing power of high-performance computers, researchers try to identify new molecules and new potential drugs able to inhibit the activity of viral proteins. It would take many years, a large amount of money, a large number of people and resources to test 500 billion molecules with laboratory experiments. This research can be accelerated thanks to high-performance computers, which allow virtual testing and allow to forecast, through numerical models, the ways by which viral proteins interact with the molecules tested. This can be done over a period of 18 months, decreasing time, cost, and personnel required. What is a high-performance computer? A high-performance computer consists of a group of interconnected computers. Working together, they perform operations extremely quickly and allow calculations which would take years for a normal computer to be performed. The power of high-performance computers lie in their ability to perform many calculations simultaneously. How can this processing power be measured? It is measured by using flops. It is the number of floating point operations performed per second. Among the high-performance computers working in the Exscalate4Cov project, there is Marconi 100, which is located in Emilia-Romagna at Syneca's headquarters. Syneca is a member of the Big Data Association. Today, Marconi 100, composed of 1,000 computers connected together with a high-speed network, is among the 10 most powerful high-performance computers in the world, second in Italy and in Europe. With a capacity of 32 petaflops, that is the ability to perform 32 million billion calculations per second, Marconi 100 is able to process up to 1.5 million molecules in a second. Is it possible to spare laboratory experimental testing? Absolutely not. In the Exscalate4Cov project, high-performance computers and classical research approaches work together in a complementary way. Virtual testing is carried out in the first stage of the project. High-performance computers process large databases of molecules and select only those which may have more opportunities to interact with viruses' proteins. In the second stage, potential drugs are tested in laboratories. This is the only way through which researchers can actually evaluate the ability of these molecules to stop the replication of the virus and therefore the progression of the disease. Eventually, the most likely to succeed molecules, which were shown to be effective also during lab testing, are tried out on patients. How are virtual tests carried out in Phase 1? At the beginning, viral proteins are investigated, their 3D structure and their behavior over time analyzed. In order to do this, molecular modeling software is used. This is able to obtain a 3D structure of a protein starting from its sequence. Since proteins are unstable and dynamic structures, able to move, their behavior is investigated through molecular dynamics software, which simulates different structures the proteins can adopt over time. The different 3D models generated in this simulation represent the protein's different structures. At the end of the simulation, these models are tested and selected for the next phase of virtual screening. During virtual screening, the ability of small molecules to interact with viral proteins is tested in order to identify, starting from very large databases, molecules which can potentially transform into new drugs. Which molecules are tested? The molecules to be tested can be divided into two categories: already known drugs on the market for the treatment of other diseases, molecules which have not been tested on humans yet, used for different purposes, not always in the medical field. During the first stage of tests, already known drugs are tested first, since toxicity to humans has already been evaluated for these molecules. Exscalate4Cov achievements may be crucial. In few months, thanks to the great processing power, the project has tested already 400,000 molecules, and among these, it identified 7,000 potential drugs which will be evaluated in laboratories. One particular molecule showed to be effective also during experimental testing: raloxifene, a drug already on sale, used to treat osteoporosis. The final goal is finding the most effective and the safest drug against coronavirus. Exscalate4Cov will conclusively give a contribution, not only to medical research, but to the entire society to win the global challenge against coronavirus and against future pandemics.